Hello, Ungoogleable. Welcome to your life. Today is beautiful. Please log in.
Teaching a computer biology, Kelly noted that IBM shifted Watson to a cloud-based system. It was originally run on a dedicated cluster, which means that any success the initial endeavor would be easier to use elsewhere. Perhaps more significantly, however, Kelly said we had to teach Watson the language of healthcare and medicine. How does one go about teaching a computer cancer biology? We talked a bit to IBM's RJ Roygaru about the process. To get the machine started, Roygaru said they took advantage of the fact that the National Institutes of Health has compiled lists of biochemical pathways, signaling networks, and protein interactions and placed them in machine-readable formats. Once those were imported, Watson's text analysis abilities were set loose on the NIS PubMed database, which contains abstracts of nearly every paper published in peer-reviewed biomedical journals. Over time, Watson will develop its own sense of what sources it looks at are consistently reliable. Roygaru told us that, if the team decides to, it can start adding the full text of articles and branch out to other information sources. Between the known pathways and the scientific literature, however, IBM seems to think that Watson has a good grip on what typically goes on inside cells. How is it going to use that knowledge? So far, most of the cancer genome sequencing efforts have focused simply on identifying which mutations are likely to be the key to driving uncontrolled cell growth. This effort has reinforced the notion that there are a handful of core genes that trigger a variety of cancers, along with a larger array that are either involved in specific cell types or in promoting specific aspects of cancerous growth like invasiveness. Although Donnell was quick to point out that discoveries in this area are ongoing, in goes data, out comes a treatment. Ultimately, Watson's not going to be used for that. Instead, it's going to be used to help design treatments. Given the results of the DNA and RNA sequencing the guys Donnell mentioned earlier, Watson will figure out which mutations are distinct to the tumor, what protein networks they affect, and which drugs target proteins that are part of those networks. The net result will be a picture of the biochemical landscape inside the tumor cells, along with some suggestions on how clinicians might consider intervening to change the landscape. Roygaru told us that Watson will be aware of two key concepts. One is that these signaling networks often have redundancies. If you block one protein with a drug, then others in the network will make up for its absence. To avoid this, Watson can suggest combinations of drugs that target multiple arms of the network. The other thing we've found is that tumors are a population of different cells, not all of which have the same combination of mutations. This heterogeneity can be identified in the sequencing results, and Watson should be able to pick drugs that can target distinct populations within the tumor. Ultimately, Watson won't do more than make a set of suggestions and provide a convenient interface for clinicians to explore the data that led to those suggestions. As Roygaru's colleague Raymindapal Singh told me, you don't want to be making the decisions, because then you're in the medical business. But, as Donnell noted,
Thank you for logging out. Today was a wonderful day. Please come back tomorrow.
Red, blue, green.
Mic check 1 2.
Congratulations. You reject the good enough. You reject words like symbolism and postmodernism. Obsolescence is not part of your vocabulary. Every night, every day, every step of the way. The diktat of fashion. The agenda of the color wheel. Gone. It takes more than a pretty face to catch your attention. I repeat. It takes more than a pretty face to catch your attention. Good. That is very good. We are source decoded. For you. For the most perfect moisturizer. The most advanced cosmetic lineup. Scientifically proven, scientifically tested. For you.
Congratulations, calibration completed.